So we have been talking about environmental factors that affect microbial growth. I think our duita class already କରିସାରିଛୁ ଏ ବିଷୟରେ. Basically, any microorganisms that grow in very extreme conditions are called as extremophiles. ଏଇଟା ଆମେ already ଦେଖିଛୁ. Basically, we are considering various factors. Through we have already looked at solute concentration. ଆମେ ଦେଖିଲୁ water activity. ଆମେ also ଆମେ ଦେଖିଲୁ pH ହେଲା. So hydrogen ion concentration ବିଷୟରେ ଆମେ last class ଦେଖିଲୁ. So today my attention will be specifically on temperature ହେଲା. Especially the syllabus there is a lot of emphasis on microorganisms that adapt to different temperature levels. I think ଆଉ ଗୋଟେ class ରେ ଆମେ oxygen level, pressure ଆଉ radiation ବିଷୟରେ deal କରିବା. ତ ଆଜି mostly ମୁଁ temperature ବିଷୟରେ ହିଁ କଥା ହେବି ଏଇ case ରେ. ଠିକ ଅଛି. So we have seen various adaptations. We have also seen the different ways how microorganisms have adapted to solute concentration, to water activity. ହେଲା. Our last class ଆମେ ଯାହା ଦେଖୁଥିଲୁ pH ହେଲା. So ଗୋଟେ ଆମେ ଦେଖିଲୁ acidophilic, that is more closer to the acidic part. ଆଉ ଗୋଟେ alkalophilic, that is more closer to the basic part. So acidophilic, alkalophilic ଆଉ neutrophilic, which is the normal range ରେ ସେମାନେ ରୁହନ୍ତି over here. So we have seen all of this. So today my attention, as I was telling you, is on temperature. So temperature is a very important factor when you see for microorganisms. Why? Because microorganisms are, I have used the word here, susceptible to external temperature. Susceptible अर्थ होची कि they are very vulnerable हैला. Because तांको पाखरे किछि mechanism नाही that they will be able to regulate their temperature, internal temperature. Amara, at least our human body we know that if we heat up, then we sweat or we lose out the water. There is some mechanism, okay, so that we are able to cool down. So heat up and cool down, we are able to moderate our temperature. Whereas for microorganisms, that is, they do not have that mechanism over here. Our temperature, जोटा भी external temperature हमारा रहुची, that is very important in another way, because that affects something called the enzyme catalyzed reaction. जहाँ भी हमारा cell रे जहाँ भी reaction होची are always enzyme catalyzed. Well, there is a protein which is the enzyme which catalyzes these reactions, and this is always affected by the temperature. This is temperature sensitive. एठी word use करछु temperature sensitive over here. So if so each enzyme has a temperature at which it functions optimally. So in this sense, say particular temperature range भितरे ठीक रे काम करे. Otherwise, there is some other effect over there. So other effect would mean that either we go in the sense it is more or less sort of thing. Okay. So at some temperature below the optimum, it ceases to be catalytic. So say जो enzyme हमारा protein अच्छे, okay, at some temperature below the optimum. So तीनटा हमें temperature हमें गोटे concept पढ़ेगा about cardinal temperatures. So 3 important temperature points. So one is the minimum, optimal, or maximum. So basically, if you go below the optimum, it does not work. Reaction As the temperature rises from this low point, the rate of catalysis increases. So low point catalytic nathila, so slowly it increases from there. So as the temperature rises from this low point, the rate of catalysis increases to that observed at the optimal temperature. So there is no catalysis, it reaches the optimal, point. जो तरह के you can see that the rate of catalysis, जो हमारा reaction रो speed था, it increases over here. Okay, the velocity of the reaction roughly doubles for every 10 degree rise in temperature. जोटा हमारा जो reaction रो velocity जो speed था तो इटा जति कि हम 10 degrees of the body चलुआ, it increases over here. But it does not go forever. There is a particular point where it actually stops over here. Okay, so when all enzymes in a microbe are considered together, okay, as the rate of each reaction increases, metabolism becomes more active. माने ଗୋଟେ particular temperature ରେ ତମେ ଦେଖିବ is the enzymes become all of the enzymes in the microbe become active and the metabolism जोटा होଉଛି microorganism ର ସେଇଟା fast हुए same case ରେ ହେଲା, so it becomes very fast at that point of time. Okay. However, beyond a certain point, further temperature increase slows the growth. Okay, and sufficiently high temperature are lethal. Right? So just imagine. Okay, so I think ମୋ ପାଖରେ ଗୋଟେ ଏଇ figure ଅଛି ମୋ ପାଖରେ, ହେଲା ଏଇଟା ଆମେ ପରେ ଟିକେ ପରେ ଆସିବା, but just imagine this is the growth rate y axis ରେ, ହେଲା y axis ରେ growth rate ତମେ ଦେଖୁଛ ଆଉ temperature ଦେଖୁଛ x axis ରେ, ହେଲା. So a point there is nothing, okay, absolutely nothing, and slowly the temperature increases. जैसा हमें देखिलु प्रति 10 डिग्री जो increase होची temperature, मतलब प्रति 10 डिग्री जो temperature increase होची, ହେଲା, so the velocity is also increasing in that way. So here it almost तमे देखुଛ exponential increase होची, पूरा exponential increase होची, ଗୋଟେ optimal point ଅଛି, तापर जेतिकी भी तमे temperature बढ़ाओ growth एते हेबो नाही, हैला, growth एते हेबो नाही, rather it decreases, हैला. So this is how basically it works over there. So beyond a certain point, further temperature increase, further temperature increase slows the growth. Okay, ଆଉ ବେଶୀ high temperature ହେଲେ, ବେଶୀ temperature भी high ହେଇଗଲେ there is cell death, ଏପଟେ death ହେଇଯାଉଛି, ଏପଟେ there is cell death ହେଇଯାଉଛି because of the high temperature over here, है ना. So high temperature ର कौन कौन effects ଥାଏ ଆମେ ଦେଖିପାରିବା. The high temperature first ଆମର denature enzymes, because enzymes are the most important part. ତମେ ଜାଣିଥିବ ଯାହା ବି ଆମେ cell cycle ତମେ biochemistry ରେ ପଢିଥିବ microorganism ରେ ହୁଏ, जो भी cell cycle हुए ସେଥିରେ enzymes are always involved, some sort of lyase or catalase or something, some enzyme is involved which actually carries forward that particular cycle over there. So if high temperature होची, high temperature ହେଲେ ସେ enzyme କାମ କରୁନି and this metabolism say case रे हुए नहीं. So it denatures enzymes as well as transport is also affected, है, so transport हुए within the intracellular extracellular जोटा भी transport हुए सेटा is also affected, as well as other proteins are also affected, and proteins are very vulnerable to temperature increase or decrease, है. So सेटा हमें टिके एटा मने रखिबा, है, high temperature increase हेले so immediately that protein will get denatured, है, denature. This is key term over here, denature, है, because protein fold, है, तमे टिके I hope protein विषय पढ़िथिबो बायोकेमिस्ट्री रे. प्रोटीन फोल्डिंग गोटे थाए, गोटे पर्टिकुलर से अल्फा हेलिक्स बीटा शीट से भलिया टिके फोल्ड होंती, आ बाय चांस यदि टेम्परेचर जब इंक्रीज होची से डीनेचर हे जो, सो से फॉर्म टा से लूज कर दबो. आ इफ से फॉर्म टा ही लूज कर देले इमीडिएटली तार रिएक्शन करिवो नाही, सो रिएक्शन कैटालिसिस से करि परिबो नाही, सो देयर इट स्टॉप्स ओवर देयर. ଆଉ ଗୋଟେ another particular effect that temperature also has on the microbial membrane, ହେଲା, microbial membrane ଉପରେ ବି इफेक्ट पकाए सो आम ऑलरेडी अलग केस रे आम देखि सारछु स्पेशली इन दी सॉल्यूट एक्टिविटी आम देखिछु is we see the plasma membrane is acting as a semi-permeable membrane to allow the solute to move out. Something it is acting as a semi-permeable membrane. तापर हम देखलो water activity रे भी plasma membrane रो काम था है. ଆଉ ଗୋଟେ ଏଇଟା ଆମେ ଦେଖୁଛୁ temperature କେମିତି microbial membrane କୁ affect କରୁଛି. So at very low temperature the membrane solidify ହେଲା membranes solidify. So this again, this also will affect the particular membranes over there. है So cellular function could be membrane affect है जाए. Very low temperature, membrane solidify. So everything basically freezes. The entire cell freezes. At a very high temperature, okay, the lipid bilayer simply melts and disintegrates. है So ଆମେ already ଏଇଟା ଦେଖିଛୁ କି protein ରେ denaturation ହଉଛି lipid bilayer ରେ କଣ ହବ it will melt and disintegrate. So again this also leads to lysis of the cell. The cell is also going to die because of the temperature. This is due to membrane ଉପରେ ଯାହା effect ପଡୁଛି microbial membrane ଉପରେ ଯାହା effect ପଡୁଛି. So that's when organisms are above or below their optimum temperature, both function and cellular structure are affected. So एठी हमर की टर्म कौन है एथिरे कि जो हमर ऑप्टिमल टेम्परेचर अछि ए ऑप्टिमल टेम्परेचर शुड ऑलवेज बी देयर. माइक्रो ऑर्गेनिजम्स मुझे अब द बिगिनिंग ऑफ द क्लास रे कहली कि इट इज वेरी वल्नरेबल से माने I mean function kariparantani if the proper temperature is not met. If the proper temperature is not met, they are unable to function at all over here. So that is the major problem that is there over here. So next we come to a very important concept, the cardinal temperatures over here. Cardinal temperature कौन कौन अच्छे? So because of the opposing temperature influences, okay, microbial growth has a characteristic temperature dependence. हम देखिलो कि बेसी लो है गले भी there is a problem, है ना? बेसी लो है गले भी there is a problem, बेसी high गले भी there is a problem. So ଆମର ଗୋଟେ particular ତିନିଟା point ରହିଛି, ତିନିଟା characteristic point ରହୁଛି which is very important for the microbial growth over here. ଏଇଟା ଆମେ first ଦେଖୁଛୁ the minimum point, ଆଉ ଗୋଟେ ଆମେ ଦେଖୁଛୁ is the optimum point, ଆଉ ଗୋଟେ ଆମେ ଦେଖୁଛୁ is the maximum point over here. ତିନିଟା point ଦେଖୁଛୁ ହେଲା. ଏଇ graph ଟା ମନେ ରଖିବ, ଏଇ graph ଟା is very important. Growth rate ଆମର y axis ରେ, temperature on the x axis, minimum, optimum, maximum ଏଇଟା ତମେ biochemistry ବି ପଢିଥିବ, but just ଏଇ case ରେ I am just reiterating that ଆଉ ଥରେ ମୁଁ emphasize କରୁଛି ଏଇ ତିନିଟା temperature, minimum, optimum and maximum. Okay, so although the shape of temperature dependence curve varies ହେଲା, so ଏଇଟା ଗୋଟେ generalized curve ଏଇଟା ଦିଆ ହେଇଛି, ଏମିତି ନାହିଁ କି ସବୁବେଳେ ଏମିତି ଦେଖାଯିବ. organism to organism ସେଟା change ହେଇପାରେ. ସେମାନେ ଆମେ ଦେଖିବା ଆମେ ଆଉ ଗୋଟେ ଗ୍ରାଫ ରେ ଦେଖିବା ଏଇଟା କେମିତି ଅଲଗା ଅଲଗା ହେଇଥାଏ ଅଲଗା ଅଲଗା organism ପାଇଁ, but ଏଇ ତିନିଟା point is constant, minimum, optimum ଆଉ maximum, ଏ ତିନିଟା point always constant ଥିବ. So although the shape of the temperature dependence curve varies, ହେଲା, the temperature optimum is always closer to the maximum than the minimum. So ଏଇ sentence ର ମାନେ କଣ ଟିକେ ଆମେ ଦେଖିବା ଏଠି ହେଲା. The temperature optimum is always closer to the maximum than the minimum ହେଲା. ତା ଅର୍ଥ ଏଇଟା ହେଲା କି ଏଇଟା ଯୋଉ ତମେ ଦେଖୁଛ ହେଲା, ଏଇଟା ଯୋଉ ତମେ ଦେଖୁଛ ଏଠି optimum ଆଉ maximum ଭିତରେ ଯୋଉ difference is very less ହେଲା. So if this is the optimum ହେଲା, ଏଇଟା optimum suppose ଏଠି ରହିଲା, so ଏଇ distance ଟା ତମେ ବୋଧେ ଦେଖି ପାରୁଥିବ ହେଲା, ଏଇ distance ହେଲା, ଏଇ distance ହଉ କି ଏଇ distance ହଉ ହେଲା, ଏଇ distance ହଉ ତମେ ଜାଣି ପାରୁଥିବ କୋଉଟା less ହେଲା. So the optimum temperature is always closer to the maximum temperature in most of the cases ହେଲା most of the cases ରେ ସେଇଟା ସେମିତି ରୁହେ ସେ case ରେ over here. Okay, କାହାକୁ କିଛି ପଚାରିବାର ଅଛି till now? କିଛି questions? Any questions? Cardinal temperature? Yes. Okay, ठीक ଅଛି. So we will continue. Alright, so ଆମେ ଏଇ ତିନିଟା ପଏଣ୍ଟ ଦେଖିଲେ minimum, optimum ଆଉ maximum. So ଆର ବି କେତେଟା ଆମେ ଦେଖିବା based on a curve ଆମେ ଦେଖିବା ଆଉ କେତେଟା curve ବି ଅଛି ଆମର. So the cardinal temperatures are not rigidly fixed. ହେଲା ମୁଁ କହୁଛି କି in the sense it differs from organism to organism. ହେଲା ସେ different organism ରେ different curve ଟା ଆସେ but this is not rigid. ହେଲା rigid ମାନେ ପୁରା exactly it's not fixed. Okay, it's not fixed. And it is always dependent on other environmental factors. Already हम pH विषय देखी सारिछु acidic environment थाईपारे कि you might have the alkaline environment. ଆମେ ମନେ ରଖିବା କି ଏଇ factors ଗୁଡା we are not going to consider them one by one. ଏମିତି ନାହିଁ କି ଗୋଟେ ଗୋଟେ ଏଇ factor act କରୁଛି. ସମାନ time ରେ solution ବି ସେମିତି ଥିବ, solute activity ସେମିତି ଥିବ, water activity ବି ଥିବ, pH ବି ଥିବ. ଆମେ କଥା ହଉଛୁ temperature ବିଷୟରେ, oxygen କାଲି କଥା ଆମେ next class ରେ କଥା ହବା oxygen, radiation ବହୁତ ଗୁଡା ଏମିତି factors ଥାଏ. So always we will need to remember that all of these factors are working together over here. So ଏଠି what we are talking about is basically the cardinal temperature is dependent on other external factors. So we have pH as well. So pH is temperature pH ଉପରେ dependent हुआ. ଆଉ ଗୋଟେ ଆମେ ଦେଖିବା it is also depending upon the available nutrients or what is available nutrients for it. Nutrients ମାନେ ତା ପାଇଁ food. Okay. ଯାହା ତାର ଯାହା ସେ କନଜ୍ୟୁମ କରେ ସେ ମାଇକ୍ରୋ ଅର୍ଗାନିଜମ ତା ଉପରେ ବି ଡିପେଣ୍ଡେଣ୍ଟ କରୁଛି. ଏଠି ଗୋଟେ ଏକ୍ସାମ୍ପଲ ମୁଁ ଦେଇଛି Eithridia fasciculata. ଠିକ ଅଛି. So this is another organism ହେଲା. So this is a flagellate protist. Flagellate protist basically it is a protist which has a flagellum for movement. And this lives in the gut of the mosquitoes. तो तमें जनरली जानथिबो प्लाज्मोडियम फाल्सिफरम विषय जानथिबो मलेरिया जोटा कॉज करे जानिछ एटा प्लाज्मोडियम लिखुछु प्लाज्मोडियम विषय पढिछ राइट यस
Yes, sir.
Okay, so you have read about the microorganism. ठीक है. So you have read about malarial microorganisms. So apart from Plasmodium falciparum, এটা ଆମେ ଆଉ ଗୋଟେ organism ଦେଖୁଛୁ that is living in the gut of the mosquitoes over here. Okay. So generally in a simple medium, simple medium could be agar medium की जो medium रे ताकू laboratory grow करंती. So it basically grows within this range 22 to 27 degree centigrade. But if the medium is supplemented with extra metals, amino acids, vitamins and lipids, it RO range change हो जाଉଛି 33 to 34 हो जाଉଛି. So you can see that so not only pH, nutrients or availability basis रे भी the microorganisms temperature range changes. So from 20 to 27 you say around maximum temperature increases to 34. So ସେମିତି ବି ସେ increase କରିପାରୁଛି over here. So ଆଉ କେତେଟା generalization ଆମେ ଟିକେ ଦେଖିବା. So optima usually range from 0 to 75 degrees Celsius. So microorganisms को ଆମେ ଯେବେ ପଢୁ ତାଙ୍କ ବିଷୟରେ so they generally always lie within 0 degree to 75 degree centigrade. ହେଲା always ସେଇଟା mostly ହେଲା. So usually ଏଇଟା ମୁଁ word use କରୁଛି usually ହେଲା. So ଏଇ 0 degree ରୁ 75 degree ଯାଏ always ଥାଏ ହେଲା. So microbial growth occurs at temperatures extending from less than minus okay minus 20 degree centigrade to over 120 degree centigrade. So ऑप्टिमल रेंज हला हमें तीनटा पॉइंट कौन देखिଛୁ एटा देखिଛୁ मिनिमम ऑप्टिमम और मैक्सिमम और डिफरेंट ऑर्गेनिजम्स को पढ़ेला बेले हम देखिଛୁ द ऑप्टिमम इज ऑलवेज बिटवीन 0 to 75 whereas -20 ରୁ 120 का अदर मिनिमम -20 जो 120 जाए सेटा है ଆଉ କେତେଟା ଆର୍କିଆ ହେଲା କେତେଟା ଆର୍କିଆ they also are surviving at 121 degree centigrade that is 250 Fahrenheit ହେଲା ଏଇଟା କୋଉଠି ଅଟୋକ୍ଲେଭରେ ବି ୟୁଜ୍ ହୁଏ ଯେମିତି ତମେ ଆଇ ଡୋଣ୍ଟ ନୋ ତମର କିଛି ମେଥଡ୍ସ ଥିଲା ନା ନାହିଁ ବାୟୋକେମିଷ୍ଟ୍ରିରେ କି ମାଇକ୍ରୋବାୟୋଲୋଜିରେ ମେଥଡ୍ସ ପଢିଥିବ ଅଟୋକ୍ଲେଭ ଇଜ ଜେନରାଲି used for sterilization. Autoclave is generally used for sterilizing your containers for sterilization ହେଲା जो भी हमारा lab equipment को autoclave करा जाए so that it becomes sterilized and that can be used but a temperature रे मध्य some archaea are able to survive over there ହେଲା so it is just to tell you ହେଲା कि the range the general range over there so हम a slider देखिଥିଲେ is how because of the presence of some nutrients some organisms are able to extend their temperature range ଆଉ ଗୋଟେ major factor ଆମେ ଦେଖୁଛୁ is the availability of water ହେଲା ଆମେ already water activity ବିଷୟରେ କଥା ହେଇସାରିଛୁ water activity ଦେଖିଲୁ last class ରେ basically if the water is combined with other elements and it is not available for use ହେଲା so if water ର availability ଉପରେ ହୁଏ ତାର cardinal temperature range depend କରୁଛି so even at the most extreme temperature microorganisms need water liquid water to grow ହେଲା so even if ଆମେ ଏଇ temperature 121 degree centigrade ବିଷୟରେ କଥା ହଉଛୁ but you need liquid water to grow over there ହେଲା so you need water to grow it so ଏଇ କେତେଟା examples ମୁଁ ଏଠି ରଖିଛି bacteria archaea protist ଆଉ fungi ହେଲା so ଆ ଭିତରୁ କେତେଟା you should always remember at least E. coli ମନେ ରଖ E. coli हो कि जो organism तुमको मने पड़ुची सेटा जेमति मने रखिबो तार cardinal temperature you should know what is the minimal temperature तार optimal कोन और maximum कोन हैना सो एटा तुमे देखि पारतो some organisms तुम देखो बहुत लो टेम्परेचर रे ऑकर करचंती केते जण बहुत हाई रे भी जाई पारचंती हैना so some are very closer to high temperature some like to go for the lower temperatures over here so एई जो हमें देखिले एई टेबल रो तार टिके एनालिसिस हम एटा करि परिबा टिके हम देखिबा so the growth temperature range for particular microorganisms usually spans about 30 degrees so basically what this means is minimum okay minimum और maximum भितर usually 30 degree centigrade gap था है usually 30 degree gap था some species like Neisseria gonorrhea causing organism एटा है ना gonorrhea have a small range है ना तांकर बहुत कम range था इसलिए high temperature से माने survive करि पारंते नहीं but केते जण को small range था others have a very wide range also है ना so this is some variation that exists that तमे भी एठी देखि पारिबो है ना तमे एठी देखि पारिबो minimum minus maximum करके एटा देखो this is almost 30 एटा भी 30 degrees है ना एटा भी almost 30-35 एमती ए भलिया रेंज रे रहउछ बट केते जण को बहुत ह्यूज रेंज था है ना सो सेटा भी तमे देखि पारिबो so the major microbial groups differ from one another regarding their maximum growth temperature. So एटा हमें देखुछु बैक्टीरिया और आर्किया रो जो मैक्सिमम तमे देखुछु 37 to 113 the 4 रो 47 एटा 15 रो 58 so मु खाली particular examples नहीं छी but एटा this is studied that the upper limit for protists is around 50 degree centigrade ए एग्जांपल रे भी तमे देखि पारिबो प्रोटिस्ट रे 47 जाय जाय छै एटा है ए ऑर्गेनिज्म का 47 जाय जाय छै बट मैक्सिमम इज अराउंड 50 degree centigrade Some fungi grow at temperature as high as 55 to 60 degree centigrade. ହେଲା ଏଟା କେତେଟା fungus ର ସେଟା growth rate ଆମେ ଦେଖିବା ଏଠି. So for example Mucorpocillus ହେଲା our range is 50 to 58 over here. ଠିକ ଅଛି. So ଏଇଟା ବି ତମେ ଦେଖିପାରିବ. So bacteria and archaea can grow at much higher temperature than the eukaryotes ହେଲା. So ଏଇଟା ତମେ easily ଦେଖିପାରୁଛ ଏଠି. So they even go 38 79 Thermus aquaticus ଏଇ organism ଶୁଣିଛ ଆଗରୁ Thermus aquaticus କେହି ଶୁଣିଛ କି ଏଇ organism ବିଷୟରେ ଆଗରୁ? ତମର ପିସିଆର କି ଏଇ ପିସିଆର କିଛି ପଢା ହେଇଛି ଟେକ୍ନିକ ପିସିଆର ଶୁଣିଛ ଆଗରୁ ପଲିମରେଜ ଚେନ୍ ରିଆକ୍ସନ?
Yes, sir.
ପିସିଆର ରେ ଆମର କଣ ଗୋଟେ ଏନଜାଇମ ଆମେ ୟୁଜ କରୁ ନା ଟ୍ୟାଗ ପଲିମରେଜ ଡିଏନଏ ପଲିମରେଜ ର ଯୋଉଟା ଆମେ ଡିଏନଏ ପଲିମରେଜ ୟୁଜ ହୁଏ ସେଟା କୋଉଠୁ ନିଆଯାଏ? ଟ୍ୟାଗ ବୋଲି କହୁଛେ ନା TAQ ଠିକ ଅଛି ଏନିୱେ ଇଫ ମନେ ନାହିଁ ଟିକେ ଯାଇକି ପିସିଆର ଟା ବି ପଢିବ ହେଲା ସୋ ସେଥିରେ ଯୋଉ ଡିଏନଏ ପଲିମରେଜ ୟୁଜ ହଉଛି ସେଟା ଇଜ ଟେକେନ୍ ଫ୍ରମ ଦିସ୍ ଥର୍ମସ୍ aquaticus ହେଲା ଏଇ ଅର୍ଗାନିଜମ ରୁ ସେଟା ନିଆଯାଏ ସୋ ଦ୍ୟାଟ ସେଇ ବିକଜ ସେଇ ଡିଏନଏ ପଲିମରେଜ ଇଜ ଏବଲ୍ ଟୁ ଆକ୍ଟ ଏଟ୍ ଅ ଭେରି ହାଇ ଟେମ୍ପରେଚର तो पीसीआर रे पढ़िथिबो तीनटा टेम्परेचर थाए तीनटा टेम्परेचर थाए सो टैग पॉलीमरेज डज नॉट गेट डीनेचर सेटलागी सेही डीएनए पॉलीमरेज टा सेही केस रे यूज हुए ओवर देयर है ना. So anyway, so just to tell you, my bacteria and archaea, they are able to grow at very high temperature, but eukaryotes ସେମାନେ କରିପାରନ୍ତି ନାହିଁ. It's been suggested that eukaryotes are not able to manufacture stable and functional organelle membrane at temperatures above 60 degrees centigrade. तो तमें केही भी जो यूकैरियोट्स को तमें पाइबनी बियॉन्ड 60 डिग्री सेंटीग्रेड है सो ए टेबल रे भी मु जहाँ देइची एटा गोटे 50 टू 58 जहाँ देइची आउ को भी ऑर्गेनिजम्स यूकैरियोट रो दे कैन नॉट सर्वाइव बियॉन्ड 60 डिग्री सेंटीग्रेड इंक्लूडिंग ह्यूमंस. ह्यूमंस आर मच लेस 37 डिग्री रे ସେମାନେ ରୁହନ୍ତି. So ଆମେ ଏବେ ଦେଖିବା तो एटा तो हमें गोटे जनरल कर्व देखुଥିଲେ एथरे है ना. सो नॉर्मल मिनिमम ऑप्टिमल और मैक्सिमम एटो नॉर्मल कर्व देखुଥିଲେ. Now I am going to move into a more specific curve for different type of microorganisms. So ए बहुत गुड़ा ना एठी देखुଥିବ तमे है ना. साइक्रोफिलस साइक्रोटॉलरेंट मेसोफिलस मेसोफाइल्स thermophiles hyperthermophiles. So different organisms रो different range रहुଛି तमे देखिबो different colors रे अछि color indicator रे मने रखिबो. So basically you can see this is the same plot है ना. So y axis रे I am having the growth rate, x axis रे I am having the temperature. So as the temperature increases growth increases, but केते लोक केते organisms range टा defined at here. So microorganisms are placed in different classes based on the temperature range. Based on the temperature range, they are classified and they are ranked in order of increasing growth temperature. So सबुठु माने in lower order रे रहउची Cyclophyllus, okay, तापर cycrotolerant, mesophiles, thermophiles, तापर hyperthermophiles हो गया है ना. So these are also representative माने एटा सबु गुडा hyperthermophiles रो एमती थाए बोली नाही एटा गुटे average निया हैछि एते है ना. So this is the general range. So हमें टिके कोन करबा is we will now study specifically about these different classes. So ଆମେ କଣ କରିବା is basically study about the psychrophiles and psychrotolerants together, mesophiles अलगा पढ़िबा और thermophiles hyperthermophiles अलगा पढ़िबा. So basically केते जण कोल्ड टेम्परेचर रे एग्जिस्ट करंती, केते जण मझे मीडियम रेंज रे काम करंती और केते जण पूरा एक्सट्रीम हाई टेम्परेचर रे काम करछंती है ना. So first we begin with the psychrophiles and psychrotolerants. So microorganisms that grow in cold environments are either psychrotolerants or psychrophilus. Cyclotolerant mane they can survive, they are able to survive at that rate. Cyclophilus mane they are able to, they like being in cold. Okay, so cyclotolerant sometimes also called as cycrotrophs ହେଲା. So either ତମେ ତାକୁ cyclotolerants ବୋଲି କହିପାରିବ ନହେଲେ cycrotrophs ବୋଲି ବି କହିପାରିବ. So they grow at 0 degree centigrade or higher. Okay, ଆଉ ତାଙ୍କ maximum temperature is 35 degree centigrade. So ଦେଖୁଛ again it is 30 degrees only, not beyond that. So ସେମାନେ 0 degree ରେ ବି survive କରନ୍ତି ଆଉ 35 degrees ପର୍ଯ୍ୟନ୍ତ ଯାଆନ୍ତି. What about Cyclophyllus? Cyclophyllus को ଆଉ ଗୋଟେ କୁହାଯାଏ as Cryophilus ହେଲା. Cyclophyllus को you can also call as Cryophilus. So they grow well at 0 degree centigrade. Okay, ଆଉ ତାଙ୍କର max optimal growth is 15 degree centigrade. ଆଉ maximum is 20 degree centigrade. ତା beyond को ସେମାନେ ଯାଆନ୍ତି ନାହିଁ ହେଲା. So they like to be in the 0 degree range itself ହେଲା. So they like to be in the 0 degree range as well. So just I have shown some diagram here. So cycrophiles, they are isolated from the Arctic and Antarctic habitats. ମାନେ ତମେ ଜାଣିଛ they are North Pole, South Pole ହେଲା. So North Pole and the South Pole, extremely cold temperatures ସେମାନେ ସେଠି ମିଳନ୍ତି ହେଲା. Cycrophilic organism ସେଠି ମିଳନ୍ତି. So oceans consist an enormous habitats for cycrophiles because 90% of ocean water, okay, is 5 degree centigrade or colder. ହେଲା, ସେମାନେ always cold ଥାଏ. ହେଲା, water is not generally hot, it is always cold over there. Okay. A particular ଗୋଟେ protist ବିଷୟରେ ଆମେ ଦେଖିବା. This is a cycrophilic protist called Chlamydomonas nivalis, can actually turn a snowfield or glacier pink with its bright red spores. So ତମେ ଏଟା Google ବି କରିପାରିବ. Chlamydomonas nivalis. तो ସେ କ'ଣ କରିବ, ଆଇଦର ସେ ଗ୍ଲେସିୟର ଆମର ମାଉଣ୍ଟେନ ରେ ମିଳେ କି ସ୍ନୋଫିଲ୍ଡ ରେ, ସେଟା ପିଙ୍କ କଲର ହେଇଯାଏ. So it becomes pink color. Pink color ମାନେ you know this is the organism is existing in that particular ସେଇ ଏରିଆରେ ସେ exist କରେ. ତାପରେ cycrophiles are widespread among bacteria, okay, and are found in genera such as Pseudomonas, Vibrio, Bacillus, Photobacterium, Shevonella. So ଅଲଗା branches ରେ bacterial species ରେ ବି ସେଟା ମିଳେ ହେଲା. ତମେ ଏଟା ଦେଖିପାରିବ ହେଲା. You can search about that, but you remember about particular examples that I have mentioned, especially the Pseudomonas nivalis that is able to survive at very cold temperature. So what we have studied is organisms that are surviving in very cold temperatures, very cold temperatures. So how do they survive? Okay, so cycrophilic microorganisms have adapted to their environment in several ways. କ'ଣ ତାଙ୍କର adaptation ରହିଛି ଆମେ ଟିକେ ପଢିବା. So first, their enzymes ହେଲା, ତାଙ୍କର we'll just see about the enzymes. Okay, their enzymes, transport systems and protein synthetic machinery ହେଲା. କ'ଣ ଦେଖିଲେ, enzymes, transport systems and protein synthetic machinery function well at low temperature. ଆମେ ଯାହା ଯାହା ପଢିଥିଲେ ଆଗରେ enzyme କାମ କରେନି କି protein denature ହେଇଯାଏ, ଯାହା ଯାହା ଆମେ previously ପଢିଥିଲେ ଏଟା ସବୁ low temperature ତାଙ୍କର they function well over here. ଆଉ ଗୋଟେ ଯୋଉ ଆମେ ଦେଖିଲେ fourth is the membrane. The membrane of the cycrophilic microorganisms have high level of unsaturated fatty acids and remain semi-fluid when cold. ଆମେ ଦେଖିଲେ in the beginning is that in colder temperature usually the semi-permeable membrane becomes frozen ହେଲା. So ସେ କିଛି move କରିପାରେ ନାହିଁ, but because they have highly unsaturated fatty acids ସେଇଟା ପୁରା freeze ହୁଏନି but becomes semi-fluid when it is very cold ହେଲା. And what else do you see? Many cycrophills begin to leak cellular constituents at temperatures higher than 20 degrees. So ସେମାନେ they cannot survive if the temperature goes up. If the temperature increases beyond 20 degrees centigrade, ତାଙ୍କର cellular membrane disruption ହେଇଯାଏ. So their membranes burst and they die. So they can only survive say sub-zero temperature or 20 degrees maximum. So ତା beyond को ସେମାନେ ଯାଆନ୍ତିନି, otherwise they die off over there. So cyclophilic organisms have adapted to their environment in several ways. So ଏଇଟା ତ ଆମେ ଦେଖିଲେ କି ତାଙ୍କର enzyme, ତାଙ୍କର transport system ଆଉ protein machinery ଠିକ କାମ କରେ। ଆଉ ଗୋଟେ ଆମେ ଏଇଟା ପଢିଛୁ already compatible solutes ବିଷୟରେ ଆମେ ପଢିଛୁ ହେଲା ଆମର when we were reading about the solute activity ହେଲା ଯୋଉଟା ଆମର କେତେର organisms ରେ compatible solutes ଥାଏ because they are trying to adapt against NaCl ସେମାନେ potassium chloride କୁ ସେମାନେ release କରନ୍ତି କି ବି accumulate କରନ୍ତି so many cyclophills also accumulate compatible solutes ହେଲା so rather than protecting against osmotic stress जोटा हमें beginning रे पढ़िथिलु in this case the compatible solutes decrease the freezing point of the cytosol this is very important ହେଲା the cytoplasm the freezing point को कमाई दिए से ହେଲା so that कौन सेटा freeze ହେବ ନାହିଁ ହେଲା and other cyclophiles also use antifreeze proteins ହେଲା this is another adaptation so there are specific proteins that are antifreeze जो माने easily freeze होती नहीं so by increasing the amount of antifreeze proteins they are able to decrease the freezing point of the cytosol so दुईटा जिनस तमे देखिबो either first ଆମେ ଦେଖୁଛୁ is using the compatible solutes ଆଉ ଗୋଟେ ହେଉଛି ତାଙ୍କର antifreeze proteins use କରିକି they are able to decrease the freezing point of the cytosol over here ଆଉ ଏଇ psychophilic bacteria of fungi are the major causes of food spoilage ଯାହା ବି ତମର fridge ରେ ବି food ଟା ଖରାପ ହେଇଯାଏ ଯାହା food ତମେ ରଖୁଛ fridge ରେ ରଖୁଛ ସେଥିରେ କାହିଁ ଖରାପ ହଉଛି because of the psychophilic bacteria ଆଉ fungus ହେଲା because they are able to exist in lower temperatures also ହେଲା so ଏଇଟା ତମେ ମନେ ରଖିପାରିବ ଆଉ ଗୋଟେ organism ଆମେ ଦେଖିବା ଏଠି is the mesophiles ହେଲା mesophiles ଆମେ ଦେଖିଲୁ ଏଇ graph କୁ ଆମେ ଟିକେ ଦେଖିବା so we have read about the psychrophiles and tolerant ସେମାନେ lower temperature ରେ କାମ କରନ୍ତି ଏବେ ଆମେ ପଢୁଛୁ about mesophiles we are reading about the mesophiles over here mesophiles basically କିଛି କହିବିନି ତା ବିଷୟରେ so mesophiles are organisms that grow in moderate temperature ହେଲା moderate temperature ତା ମାନେ କ'ଣ optima is from 20 degrees to 45 degrees and often have a minimum 15 to 20 and maximum of 45 over here ହେଲା so easily ତମେ ମନେ ରଖିପାରିବ ଏଇଟା 15 degree centigrade ରୁ ହେଲା so basically if you see the curve so 15 degree centigrade maximum 20 ରୁ 45 ହେଲା तो ये भलिया कौन गोटे कर्व हेबो तापर 15 ରୁ 45 ଭିତରେ काम करे and हमको एटा गोटे मन रखि परिबा is almost all human pathogens are mesophiles एटा मन रख उत्तरा हमर human pathogens था जो माने हमको harm करंती pathogenic organisms are mesophiles so as might be expected because the body is constant at 37 degree centigrade है ना so that is why हमको ज्वर आसे काहे ज्वर आसे because the body increases the temperature beyond 39-40 degrees per so that a mesophile mane they die okay so that is the purpose the purpose of having high temperature तुमको ज्वर से लगे हो because the body is doing the fight है ना body is increasing the temperature to kill the mesophiles over there so mesophiles वगैरह बेसी नहीं मो एतिकी खाली कहि last before we close ଆମେ ଟିକେ କଥା ହେବା about thermophiles ହେଲା so lower temperature ଆମ ବିଷୟରେ ଆମେ କଥା ହେଲେ about high temperature medium temperature କଥା ହେଲେ ଏବେ ଆମେ କଥା ହେବା with about high temperature so microbes that grow best at high temperatures are thermophiles or hyperthermophiles so ସେମାନେ କ'ଣ କରନ୍ତି thermophiles grow at temperature between 85 to 45 to 85 degree centigrade ଆଉ ତାଙ୍କର optimal temperature ଥାଏ between 55 to 65 degree centigrade so this is the temperature range where they exist ଆଉ ଏଇ range ରେ ତମକୁ ये मिलिबे नहीं माने eukaryote organism तमको मिलिबे नहीं ହେଲା so usually what you have is the bacteria ଆଉ archaea bacteria or archaea a few photosynthetic protists and fungi are thermophilic आउ केही भी एथिरे यूकैरियोट तुमको मिलिबे नाही हैला सो these are the microorganisms that usually find ଆଉ thermophiles ର habitat କଣ କୋଉଠି କୋଉଠି ମିଳନ୍ତି thermophiles flourish in many habitats composts ରେ ହେଲା ଆମର ଯୋଉ କଣ ହୁଏ ସେଟା ଆମର ଯୋଉ ତମେ ଡିକେ ଯୋଉଠି କରୁଛ ମାନେ ସବୁ ଡେଡ୍ ଲିଭ୍ସ ଆଉ ସେଟା କମ୍ପୋଷ୍ଟ ଜେନେରାଲି ତମର ଖତ ଯୋଉଠି ଥାଏ କମ୍ପୋଷ୍ଟ self hitting haystacks so ତମ ମାନେ because ତମେ ଏବେ ଘରେ ଅଛ you are able to uh, ମାନେ ତମ you are in an area where you can easily find them so composts ହଉ କି self hitting haystacks ହେଲା ଯୋଉ ଆମର ମୁଁ କହିପାରୁନି ଓଡ଼ିଆରେ haystack କୁ କଣ କୁହାଯାଏ ମତେ ପାଳ କୁହାଯାଏ ମୁଁ ଜାଣିନି ଠିକରେ କହିବ ମତେ ହେଲା ସୋ haystack ଯୋଉଠି ବେଶୀ ଜମା ହେଇଥାଏ ସେଠି thermophiles ମିଳନ୍ତି hot water lines ହେଲା ଆଉ hot springs ଯୋଉଠି ଏଇ କେସରେ ସେମାନେ ବହୁତ ମିଳନ୍ତି ଘରେ ହାଇପର ଥର୍ମୋଫାଇଲ୍ସ ଯୋଉଟା ଥାଏ ଟିକେ ମିଡିୟମ ରେଞ୍ଜର ତ ହାଇପର ଇଜ ମଚ ମୋର ହେଲା ସୋ ହାଇପର ଥର୍ମୋଫାଇଲ୍ସ have growth optima between 85 to 130 ହେଲା so uh, they have a they grow between 85 degrees centigrade to 113 degrees centigrade so they are hyperthermophiles the temperature range both high ଥାଏ so they usually do not grow below 55 degrees centigrade ମାନେ ତମେ ଭାବ ତାଙ୍କର ଏଇ graph ତମେ you see so their minimum is 65 55 or above ହେଲା it is above ଆଉ ତଳେ they don't exist within the ଏଇ range ରେ ସେମାନେ exist କରନ୍ତି ନାହିଁ they are starting from 55 to 60 to 100 ଉପରକୁ they go for so that is how thermophiles work ଦୁଇଟା ଏକ୍ସାମ୍ପଲ ମୁଁ ମେନସନ କରିବି is Pyrococcus abscissi and Pyrodictyum occultum ହେଲା so ଏଇ ଦୁଇଟା organisms ଆମର ଅଛି so these are examples of marine hypothermophiles and are found in hot areas of the sea floor ହେଲା ଏଇଟା हमको sea floor ରେ ମିଳେ ଯୋଉଟା ବହୁତ volcanic ଜାଗାରେ ବି ଏଇଟା हमको ମିଳେ so these are hyperthermophiles over there ହେଲା hyperthermophiles so how do the hyperthermophiles adapt to their environment? Okay, how do the hyperthermophiles adapt to their environment? So first ଆମେ ଦେଖିବା is they have heat stable enzymes. Okay, they have heat stable enzymes and protein synthesis systems ହେଲା so ଏ ଦୁଇଟା ତାଙ୍କର mechanism ଅଛି that function properly at high temperature ହେଲା mainly ଆମେ ଦେଖୁଛୁ ସେଇ enzyme ଆଉ ସେଇ protein ହେଲା କେତେ କେତେଟା organisms ର low temperature ରେ କାମ କରେ କେତେଟା organisms ର high temperature ରେ କାମ କରେ so these proteins are stable for a variety of reasons ହେଲା heat stable proteins have highly organized hydrophobic interiors, okay, and more hydrogen and other non-covalent bond to stabilize the structure. So, এটা ତମକୁ research ରେ ପରେ କାମ ଆସିପାରେ is basically co-protein heat stabilize ଥାଏ ହେଲା so which protein is able to survive at high temperatures. So ତାଙ୍କର କ'ଣ କ'ଣ characteristics ଥାଏ is they are having hydrophobic interiors ହେଲା more hydrogen ଆଉ more non-covalent bonds to stabilize their structure. So this is how a high heat stable proteins are characteristic. Large amount of amino acids such as proline, ହେଲା ଏଇଟା ଆଉ ଗୋଟେ characteristics also make the polypeptide chain less flexible and more heat stable. So ଏଇଟା ଆଉ ଗୋଟେ factor ଆମେ ଦେଖୁଛୁ ହେଲା. In addition, the proteins are stabilized and aided in folding by proteins called chaperone proteins. It ଆଉ ଗୋଟେ class of proteins that help in protein folding and especially high temperature they are able to help in protein folding chaperones ହେଲା so these are the ମାନେ କ'ଣ କହିବି reasons why ତମର protein exist କରିପାରୁଛି high temperature heat stable protein ର characteristics ଗୋଟେ ହେଉଛି hydrophobic interior ଆଉ ଗୋଟେ ହେଉଛି more hydrogen ଆଉ ଗୋଟେ ହେଉଛି non covalent bond ବହୁତ ଗୁଡ଼ା ତାଙ୍କର ଅଛି okay and ଆମେ ଦେଖୁଛୁ proline ବହୁତ ଥାଏ ତାଙ୍କର okay and chaperone proteins ତାଙ୍କର ଥାଏ for heat adaptation ତ ଏଇଟା ତ ଆମେ କଥା ହେଲୁ protein ବିଷୟରେ this is mainly about the proteins okay this is mainly about the proteins But ଆଉ ଗୋଟେ ଆମର ଦେଖିପାରିବା ଆମେ DNA ବିଷୟରେ. We can talk about the DNA. So nucleoid assisted proteins appear to stabilize the DNA of the thermophilic bacteria. So high temperature ରେ ତାଙ୍କର DNA disrupt ହୁଏ ନାହିଁ because they have this nucleo associated proteins ହେଲା ଯୋଉଟା nucleus ସାଙ୍ଗରେ ଲାଗିଥାଏ. ଆଉ ଗୋଟେ ଆମେ ଦେଖିବା is hyperthermophiles have an enzyme called reverse DNA gyrase. Reverse DNA gyrase that changes the topology of the DNA and enhances its stability. So it a DNA ର survivability. So ଗୋଟେ ହଉଛି ତମର nucleo assisted proteins ଆଉ ଗୋଟେ ହଉଛି DNA reverse DNA gyrase ହେଲା. So ଏଇ ଦୁଇଟା adaptation ପାଇଁ they are also able to survive over there. Third ଏଇଟା ଆମେ କ'ଣ ପ୍ରୋଟିନ୍ ଦେଖିଲେ ଏଠି ଦେଖିଲେ DNA ଏଇ କେସରେ ଆମେ ଦେଖୁଛୁ membrane. This is the membrane adaptation. Membrane ରେ କ'ଣ ଦେଖୁଛୁ membrane lipids of thermophiles hypothermophiles are also quite temperature stable. କେମିତି ହେଇପାରୁଛି so they tend to be more saturated, more branched and of higher molecular weight है ना higher molecular weight ये तीनटा characteristic करता है more saturated more branch और higher molecular weight था so this increases the melting point of the membrane lipids है ना so एटा तांकर membrane adaptation okay so archaea have membrane lipids with ether linkages एटा बस archaea रे generally एटा 4 लिखुची but so such lipids are resistant to hydrolysis at high temperatures. Okay, ଆଉ ବି ତାଙ୍କର ଏମିତି ଥାଏ diglycerol tetraethers are observed in some archaeal thermophiles. Okay, and they also provide some rigidity. Okay, so ଏଇଟା ହେଉଛି membrane adaptation. So ଆମେ ଦେଖିଲେ protein adaptation, DNA adaptation ଆଉ membrane adaptation. So ଏଥିରେ ମୁଁ stop କରିବି ଏଠି ତ ମୋର 5ଟା question ଅଛି ମୁଁ ଯାହା ବି